So, Dr. Spencer, I wanted to ask you about the process for procurement and how the postnatal donations are processed, because I know it can be a very intricate scenario.
What's exciting about the way we procure our healthy donations now is what is possible with the live cell count and the health and the regenerative potential of these things because of the way we're doing it and the fact that we have the ability to be in control of every step in the process, which is unique to us. As far as I know, we are unique in the ability to be able to control from the very moment that the doctor proposes to the mother that there's a potential use for the donation, besides having it discarded, to the point where we cryopreserve it and then thaw it out and have a live cell count that is remarkable. And frankly, compared to what was possible 10 years ago, honestly, it would have been unbelievable at the time. So that's what is so unique about what we do, and it's why we try to do every step of the process here from start to finish.
So I think it's important to recap that you basically control the whole process as far as working with the OB offices, the moms themselves, the hospitals, and nothing is outsourced and left to chance or other possible errors in the whole scenario. In addition, I do want to drill down a little bit deeper. Can you talk about some of the improvements in the technology that you use to all medical processes and things advance over the years, and this is no different. So can you speak to some of those differences and how you're able to achieve a better result with those donated after those donated procedures?
Yeah, great question. The first thing that was necessary was, just as you said, us as a group, a company, taking over every step in the process. And until we were able to do that, we weren't able to make the tiny improvements that we've made over the last 10 years in every facet of every part of the process. And it's multifaceted. From the time that our own people go and pick up the postnatal donation at the time of the procedure from a local hospital with its short driving distance from our laboratory, to the way that we package it and we instruct the doctors, not only with how they screen the donors, because it's actually very rigorous selection process. We don't accept donations from just anyone, to the point that we're actually immediately processing, and the way we're processing it and the steps of the processing, the reagents that we're using, the methods that we use to cryopreserve the product have improved in the last 5 years alone, that have been able to increase and improve our cell viability between 10 and 15% over the last 10 years from where they were 10 years ago. Those being able to be involved and have our hands on every step in that process have made a huge difference in the final outcome.